All right, we're going to continue our discussion of going beyond Mendelian genetics and look how things are actually a little bit more complicated than what Mendel had discovered. Now, again, none of this shows that he was wrong about anything. The law of independent assortment still applies, and the law of segregation still applies. Now, one thing here that Mendel was lucky about is that all of the genes that he studied were on different chromosomes. And so everything he studied was unlinked, and independent assortment did indeed apply to all of those. But we know that chromosomes are very large, and that there exists on those chromosomes more than one gene. So for example, if we take a look at chromosome 21, one of the smallest human chromosomes, it's made up of 50 million bases, and there's between 200 and 250 genes there. Now, depending upon the allele of the various genes, these 200 to 250 genes, there are several diseases that are related to the various alleles of the genes on this chromosome. For example, we already know that trisomy 21, so if you have 3 copies of this chromosome, that's Down syndrome. There is alleles involved in long QT syndrome, that's a heart condition, T-cell lymphoma, that's involved in a type of cancer. And so there's a lot of different diseases that are associated with alleles of some of the genes on chromosome 21. And every single one of these 200 to 250 genes are linked because they're on the same chromosome. Now, linked genes do not assort independently. Linked genes are going to be forced to the alleles of those genes to be inherited together. So let's take a look here. So we have a diploid organism, and what we're looking at here is these 2 chromosomes are not replicated. These are 2 homologous chromosomes. Okay, so be careful. I do not have a centromere there. These 2 are not sister chromatids to each other. And so what we have here is this person is homozygous for the A allele and homozygous for the capital B allele. And here's another individual that we're crossing here. This is our parental generation or our P generation. And this person is homozygous for the little a and homozygous for the little b allele. So again, these aren't sister chromatids, these are homologous chromosomes, right? They haven't been replicated yet. Now, when you cross these two individuals together, you are going to get 100% of the F1 generation, or the first filial generation, are going to be dihybrids. Big A, big B, little b. And the 2 homologous chromosomes, you know, because of what the parents were, are going to be a big A, big B chromosome and a, little b chromosome. You know that, okay, because of the way in which you set up the crosses here. Now, you're going to do a test cross, and what that means is you're going to take this dihybrid and cross it back to a double recessive individual. Little a, little b, little a, little b. Okay, now, that means the only type of eggs or sperm that this individual can make is just a, little b. And this individual here can only make a big A, big B sperm and a, little b sperm because they are on the same chromosomes. You cannot have a little a and a big B sperm because that would entail, right, breaking things up here. They are linked. So what you would expect to see then is only 2 phenotypes coming out of this cross. A phenotype that is identical to that parent, and a phenotype. Now, when I say phenotype, I'm saying the big A, big B phenotype and the little a, little b phenotype. The genotype, of course, is the dihybrid or the double recessive. Be the genotype. So you have the phenotype of one parent, you have the phenotype of the other parent. What you do not see is a recombination of those phenotypes. You do not see a big A little b phenotype or a big B little a phenotype, right? You're not seeing the results of these sperm, of these what are called recombinant sperm leading to recombinant offspring. Why? Because the genes are linked. They're going to travel together into the different zygotes. Now, Thomas Hunt Morgan was the big player here in figuring out and understanding the idea behind linked genes. And he did it studying Drosophila melanogaster, which is the fruit fly, which is a fantastic model organism because they have high generation times, and you can raise hundreds and hundreds of them and get lots of interesting results very quickly. Now, Drosophila geneticists don't use Mendelian nomenclature. Instead, what they do is they look at a particular gene and they look at the wild type, and then they look at the mutant version. Wild type is the version of the allele, the phenotype that you see most often within the wild. That's going to be red eyes. And then they find the mutant, right? The rare mutant. That's white eyes. And so they name the gene for what happens when you mutate it. So they call it white. This is the white gene. And then if it's the wild type allele, right, the allele that gives rise to the wild type phenotype, they put a little plus superscript. And if it's the mutant allele, they don't. Okay, so W plus is red eyes, W is white eyes. So what Thomas Hunt Morgan did is he set up a cross very similar to that abstract cross I showed you at the beginning of the lecture. And what he did was he set up, here is our dihybrid fly, or excuse me, our homozygous wild type fly. And here is our homozygous recessive fly. Now the two genes he was looking at is called the black gene, that's B plus and B. So the B allele gives rise to the black body, the B plus allele to the wild type sort of gray body. And the other gene is the vestigial gene. Now VG plus gives rise to normal wings, and VG gives rise to vestigial wings. They look like this, and these flies are kind of cool. They can't fly, and they look like little ants that are crawling around. Cross these guys together, and now you've got yourself your dihybrid cross, knowing that you only have two types of chromosomes. You've got the B plus VG plus chromosome, and you've got the B VG chromosome. And then you test cross. You cross that back to a homozygous recessive fly. Right? So the only chromosomes they have is little BVG and BVG, that's it. So the question is, what results do we expect to see if these two genes, the body color gene and the vestigial gene, if they are not linked? Well, let's take a look at that. Let's make a Punnett square. Now, the only type of sperm that this individual here can make is a BVG sperm. That's why they do a test cross. That's why they go against a homozygous recessive. It makes it really easy. There's only one kind of sperm this individual can make. And then, since we're assuming that these genes are not linked, then independent assortment occurs. So we apply both of the laws. The law of segregation, which states that each one of the eggs can only have one of the two alleles of each gene. And then independent assortment tells us that each allele is going to be in the same egg as both of the other alleles, so at equal opportunities. That probably didn't get explained as well as it could, so let me instead say this, that what independent assortment tells us is that it's just as likely as a B plus VG plus egg as it is a B plus VG egg. It's just as likely that we have VG with a B as it is VG plus with a B. Okay, so we have both independent assortment going, we have segregation going because these genes are not linked. Which means because each of these eggs are of equal probability, then when you do the crosses, each one of these genotypes and therefore each one of these phenotypes is of equal probability. A 1 to 1 to 1 to ratio. If you had counted a couple thousand of these flies, you would get something like 575 to 575 to 575 to 575. Okay, let's look at the phenotypes and let's break them down into what's called parental phenotypes and recombinant phenotypes. These two guys here, wild type wings, wild type color, that's one of the parents. Right, so that's a parental phenotype. This guy here, mutant wings, mutant body color, that's the other parent. So those are called parental phenotypes or parental offspring. Now look at these two. These are recombinant offspring because it's a recombination of what you see in the parents. Right, this one here is wild type body, vestigial wings. Right, that's neither of the parent. That's a combination of the parents. This one here is black body, wild type wings. Again, that doesn't exist in one of the parents, so that's called a recombinant offspring. Now, you know that the genes are not linked when the percentage of parental offspring that you see is equal to the percentage of recombinant offspring from a pair such as this. Okay, good. Now, let's take a look at what would happen if these two genes were instead linked. So we do the same cross, right, to establish our dihybrid. Then we do our test cross. And so let's take that abstract thing that we had before and apply it here. I just replaced the A's and B's with B pluses and VG pluses. So here's that cross, that double homozygous, double dominant, double recessive, to give us our dihybrid, where we know one of the chromosomes is B plus VG plus, the other one is BVG, and then we test cross it here. And so if the genes are linked, what you would expect is to only see parental phenotypes. 50% of them should look like one of the parents, 50% of them should look like the other. 50% should be double homozygous, or excuse me, double dominant, and the other one should be double recessive. Let's see what we get. Well, when we do this, here's the two parental phenotypes. And when you look at all the numbers, they sure look to be the most represented, right? So that partially matches what we want. But, right, theoretically, because these genes are linked, we shouldn't see the recombination occurring. We shouldn't see a BVG plus or a B plus VG. And yet, look at that, we are seeing a VG plus B and a B plus VG. We are seeing recombinant offspring at very low frequencies, at much lower frequencies. So we know that the genes are linked. Okay, so this does show us that the genes are linked because the percentage of parental phenotypes that we're seeing are not equal to the percentage of the recombinant phenotypes, right? And that they have to be about equal for it to be non-linked. So we know we have linkage, but got something else. We got something weird going on over here. Well, what is this? These recombinant offspring that we're seeing, despite the fact that the genes are linked, we're getting recombination. Remember, during prophase 1 of meiosis, when those homologous chromosomes pair, right, in the production of eggs from this individual here, we're getting crossing over occurring. And that crossing over, right, here's our VG plus, B plus, here's our VGB, right? So this is after DNA replication occurs during prophase 1 of meiosis. We get crossing over, we get recombination, and now look at this. We get a new chromosome being formed as a result of this crossing over, a VGB plus chromosome and a BVG plus chromosome. So now when this sperm fertilizes this egg, or this sperm fertilizes this egg, we're going to get the recombinant offspring coming out. But since this crossing over event does not happen very frequently, We don't see very many of these flies, certainly not in equal probabilities to the other two types. Now, that is an interesting number. The number that we see, 206, 185, that basically tells us just how far apart the two genes are from each other on the chromosome. The farther apart they are on the chromosome, the more likely a crossing over event will occur between the two of them. And the more often you will see this recombination occur, and therefore the higher number of recombinant offspring you will see. The closer the two genes are to each other, the less frequently you will see crossing over occur, the less frequently you will see these types of eggs, and the less frequently you will see these types of recombinant offspring coming about. And so early on, before we could sequence DNA, right? So back in the 1950s, they were mapping genes onto chromosomes by simply doing lots and lots of crosses and seeing what was the recombination frequency. And that could give them a number to tell them how far apart two genes were on a chromosome. How do we determine recombination frequency? Well, you do the cross like we showed, that test cross. And you count up the total number of offspring. So 965, 944, 206, 185. That's going to be your denominator. Then you identify the recombinant offsprings, the offspring that have a recombination of the two parental phenotypes. And you add those up, 206 plus 185. And that is your numerator. So you do the division, multiply by 100, and that gives you the recombination frequency. That gives you a sort of number that tells you a relative distance that the two genes are apart from each other. Now, it doesn't mean that they are 17 base pairs apart from each other. It doesn't mean that they are 1,700 base pairs apart from each other. It simply means that they're far apart enough where 17% of the time you will get a crossing over event occurring between those two chromosomes. And when you do this then, when you compare two genes that are all map on the same, you can start to make genetic maps. So you know, for example, that these two guys are BN and CN about 9% from each other, and that when you put CN and VG, Cinnabar and VG, you do those crosses, they're about 9.5% of each other, and then VG and B, you know, about 18 or so percent each other from apart from each other. That's how you can start to map genes on a chromosome, by simply doing crosses and seeing and counting the number of recombinant offspring that you see. Okay, now next, the characters that Mendel studied appeared equally within the males and female offspring, but that's not always the case. There are some traits, there are some characters that are linked to sex determination genes that are on the same chromosomes that determine sex. So, for example, in humans, we have the XY chromosomes are the sex chromosomes. If you have an X and a Y, you are male. If you have two X's, you are female. And the two chromosomes, the X and the Y, although look very different from each other, in fact, the X chromosome is much bigger. There's about 1,000 genes on there. The Y chromosome only have about 78 genes. Most genes, therefore, most sex-linked genes are X on the X chromosome, are X-linked. And so when we talk about sex-linked genes in this class, we're talking about genes that are on the X chromosome. Now, we need to if we're talking about a gene that resides on the X chromosome, we need a new way of showing that. So what we do is this: we make an X, boom, boom, because we know that gene is on the X chromosome. Now, if it is an unaffected female, if we're talking about a dominant, a gene that has a dominant allele and a recessive allele, like hemophilia, for example, blood disease. If it is an unaffected female, she could be homozygous dominant, like this, XH, XH. Or she could be affected female, so X little h, X little h. Or she could be a carrier, XH, big H, X little h, right? Because females have two X chromosomes. Now males are, we need to bring up a new word here because males only have one X chromosome. So we can have a male that's affected, X little h, and we call that person is hemizygous, recessive, hemi, because it only has one X chromosome. Or we can have an unaffected male, X big H, Y. We say that male is hemizygous dominant and is unaffected. So let's do a demo question. So let's take a look at a situation where we have this problem. Red-green color blindness is caused by a sex-linked recessive allele. And by sex-linked, again, we mean X-linked. If we take a color-blind man and marry him to a woman with normal vision whose father was colorblind, what's the probability that we'll have a colorblind daughter? What's the probability that we'll have a colorblind son? So let's draw that out and take a look at it. Okay, so in order to solve genetics problems, what you must do is break down each part of those sentences and extract as much information as you can get from each sentence before moving on to the next. So first, it says red-green color blindness. Okay, so we have red-green colorblind. Well, let's stop there and let's assign a letters to it. So let's call. So let's make it B. So we have two alleles, big B and little b. Now I don't know which is which yet. I don't know whether colorblindness is recessive or dominant. So we're not going to assign alleles or traits to these alleles yet. Is caused by a sex-linked recessive allele. Okay, there's a lot to unpack there. So first, it's sex-linked, meaning these two alleles are on the X chromosome. So we can't just say big B and little b anymore. We have to say X big B and X little b, right? We've got to do that. Furthermore, now we can say it's recessive. So we can say that X big B gives rise to normal vision, and X little b is colorblind. Okay. Next, all right, so we've got our alleles all figured out. Next, a colorblind man. Boom, let's stop. This is a man. So we know we have X, Y. Furthermore, he's colorblind, so we know he must have the little b allele. That was easy. All right. Marries a woman. Stop. She's a woman. Okay, so let's have that. X, X. With normal vision. Stop. Okay, she has normal vision, which means she must have, but we don't know on this other guy, right? That she might be a carrier or she might be homozygous normal, right? So marries a woman whose father was colorblind. So this woman's father, so father meaning XY, was colorblind. Which means that he could not have, the only thing that her father could have donated to her is the small b allele. So that must be her genotype. All right. Now we're good. Now we have the mom, we got the dad, we've got all the alleles fleshed out. Let's just go straight to a Punnett square, and then we'll tackle the questions. So the types of sperm that can be made is two. It could be an X sperm and it can be a Y sperm. And the two types of eggs can be an X big B egg and an X little b egg. And so we do that, we do this, we do this. And so the possible genotypes that we can see with these two parents are a X big B, X little b, X little b, X little b, X big B Y, and X little b Y. So there are 1, 2, 3, 4 possibilities. Let's see what we're looking for. What is the probability that they will have a blind daughter? Now, this is really important to understand. Okay, language is everything in these sorts of problems. And what this question is asking you is, it's saying, they don't have a kid yet. We have no information about their kid. What we want to know is what's the chance that they're going to have a colorblind daughter. Notice that's different than saying, okay, they have their daughter. What's the chance that she's colorblind? Those are two very different ways of expressing two very different concepts, okay? So, what is the probability that they will have a colorblind daughter, meaning they don't have a kid yet, they have no idea the sex of the kid or the state of that kid's vision. So, all of these are still possible. All 4 of these are possible. So what's the chance they're going to have a colorblind daughter? Well, there's a daughter, but that's the colorblind daughter, so it's going to be 1 out of 4. 1 out of 4. Now, let's think about this. Let's think about if we were to ask the question instead, like, of their daughters, what's the chance that she would be, of their possible daughters, what's the chance that one will be colorblind? Then it's different because they're giving us different information. They're saying of their daughters, meaning this becomes irrelevant. We have information telling us that this is irrelevant because we're asking only of the daughters. What's the chances? Well, that would be now it's out of 2 because that extra information is allowing us to cross this off. And which of these 2 or how much of those 2 satisfy what we want? Well, only this is affected. Only this is a colorblind daughter, so 1/2. So be very careful about how the question is worded. Okay, what information is it telling you up front? Which of these can you cross off as possibilities? Now the other is, what is the probability they will have a colorblind son? Well, there's 4 possibilities, 1, 2, 3, 4, because of the way they worded the question. And colorblind son is just that 1 there. So 1 out of 4 will be a colorblind son. So that is how you work on solving a problem like this. Pick apart each part of that sentence, of that paragraph, of that question, and pull out all the information you can before trying to solve the problem. Okay, finally, one interesting complication about X chromosomes in females. In all species where the female is designated by 2 X chromosomes, early on in the development of the embryo, of with the 2 X chromosomes, randomly, each cell within that embryo, one of the 2 X chromosomes is going to be inactivated. Its chromatin is going to be packaged so tightly that within a light microscope, you can see that X chromosome as what's called a bar body. So the bar body is the inactivated X chromosome. Now, in some animals, you can actually detect this random X inactivation. For example, cats, right? Your tortoiseshell cats always have to be female. Why is that? Because the gene for fur color is on the X chromosome. And there's an allele that gives rise to orange fur, and there's an allele that gives rise to black fur. And early on in development, when each of the X chromosomes randomly becomes inactivated in the cells that will give rise to hair, what you'll have is, well, if this cell, the X chromosome with the black allele was inactivated, become an R body, then all the hair that come from this cell are going to be orange. And so there's your patch of orange. Then randomly another cell in which the orange allele was inactivated due to X inactivation, only the black allele was being expressed, then all the hair that derives from this cell will be black. And so now there's your patch of black. So this sort of mosaicism that you see here, that in different cells, only one of the two X chromosomes is randomly active and the other one is inactivated, you'll see that as this fur color, this change in fur color. Okay, so that is the end of our discussion on genetics. That's the end of Bio 180 here. What I do want to say is genetics is not easy. Okay, genetics is not something you can watch a lecture and then say, okay, I got it. You've got to practice solving problems. There's problems on the Mastering Biology. There's problems in the end of the chapter in the book. There's lots of potential there for you to practice. You've got to do those. It's like you'd never sit and watch a math lecture without ever doing a math problem before taking an exam. You've got to practice taking those problems.